Hello everyone. In this video we are going to study about the formation of hemoglobin. As the name suggests, it is formed of heme plus globin. So hemoglobin synthesis starts in the pro-erythroblast stage, and as we know about the pro-erythroblast stage that we studied in the formation of the blood series, erythropoiesis. Uh, So how is it formed and what are the components of this molecule heme? So we are going to study about heme first. So from Krebs cycle, as we all know where the Krebs cycle occurs, it occurs in the mitochondria of the cell. So from the Krebs cycle we get this succinyl CoA, and succinyl CoA combined with this glycine forms a pyrrole molecule. Then four pyrrole rings combine together to form a protoporphyrin 9, and that combines with the iron Fe2 plus and forms the heme. Now, this heme, when combined with the polypeptide chain, also known as the globin chain, forms the hemoglobin. So, how is it formed? From succinyl CoA plus glycine to pyrrole molecule, then four pyrrole rings together to form protoporphyrin 9th. With iron, it forms heme. And heme with globin or polypeptide chain, it forms hemoglobin. Now, this polypeptide chain could be of different types like alpha, beta, gamma, and in adults we know that this hemoglobin A is very common in adults, and in fetus hemoglobin F is very common. So this hemoglobin A is formed of two alpha chain and two beta chain. Now one question that is probably asked in the MCQs is that how many iron molecules are present in one hemoglobin molecule. Most of the students write it as one iron molecule, but now we know that hemoglobin have two alpha chains, two beta chain, and one chain has a one heme molecule. One heme molecule has one iron. It means in one hemoglobin molecule there are four iron atoms. Okay, four iron atoms present in the one molecule of hemoglobin. Now, how hemoglobin combined with the oxygen O2? So there's a what kind of combination it is? It is a very loose combination. Where it is, it's a very loose combination, plus it's very reversible. Loose plus reversible, and bond is a coordination bonds of iron. It form a bond with the coordination bonds of iron atom. It does not combine with Fe2 plus. Oxygen do not combine like this, like two oxygen molecules will combine like this and form FeO2. So it doesn't happen. They do not form ionic bond. There is a very loose and reversible bond, kind of a coordination bond of iron atom, bonding with the coordination bonds of iron atom. So why it is loose? So that in the tissues it can dissociate, and tissues can get proper oxygen, and it can bind easily, and it can reverse easily. That's why there's a very loose and reversible combination or the bonding. Now we'll study about the iron metabolism. That is a very major part of biochemistry also. Biochemistry and minerals that will study later, vitamins and minerals. So let's look at some information about iron. So body average of iron is 4 to 5 grams. 65% of iron is present in hemoglobin, 4% as myoglobin, 1% various heme compounds. There are other heme compounds also. 0.1% protein transferrin, that will study how iron gets transferred. So 0.1% is bind to apotransferrin to form the protein transferrin, and 15 to 30% ferritin, which like iron combines with apoferritin to form ferritin, and iron also combines with apotransferrin to form transferrin. So how it is absorbed? Let's have a look in this diagram first. uh, First of all, like that, we ate the food and food contains iron, it gets absorbed. One most important thing that we have to remember it, that it gets absorbed in Fe2 plus form, not in Fe3 plus form. Does not get absorbed. Fe2 plus form is only getting absorbed from the intestines, small intestine. Now this iron that got absorbed, it combines with this apotransferrin and forms transferrin. Now this transferrin will get converted to ferritin, or it can form hemosiderin, or it can form heme also. One more important thing, that ferritin is a kind of storage molecule. It stores the iron in it, and heme helps in the formation of hemoglobin. Uh, from where this apotransferrin came? It came from the liver. Liver synthesized this apotransferrin, is synthesized by this liver, and it gets how it reaches here. It reaches here with the help of bile duct. It get mixed with bile, and it reaches here in the blood through bile duct toward in the periphery of the intestine. Now let's look at this chart. So there's this free iron. That free iron is getting converted to ferritin. Ferritin is a storage iron. Hemosiderin, it is a very insoluble form. Then heme and enzyme. One more important thing that we have to remember is, this is reversible, this is reversible, this is irreversible. This is not reversible. Like when this hemoglobin will get digested or it get metabolized, then it will get converted to free iron again, not directly. It can also get convert, it can also bind to transferrin. So from transferrin we can get free iron, or from uh, free iron we can get transferrin, bond with iron molecule. Now this can form hemoglobin, red cells. This heme can come here, or there are some other heme containing compounds. Blood loss, 0.7 milligrams iron daily in the menses. Fe2 plus absorbed in small intestine. Fe excreted, zero point 0.6 mg daily. Now this hemoglobin, degraded hemoglobin, free iron, macrophages, bilirubin, that this part we'll study later. That is a very important part for jaundice also. Next we have destruction of hemoglobin, that we were studying about in very crisp form. Now this is a very detailed form. So like first, this hemoglobin will get burst. So how this hemoglobin is going to get burst? Actually the red blood cells we have, now this is a blue blood cell. The red blood cells we have, these red blood cells have certain enzymes. These enzymes helps in maintaining the integrity of their cell membrane. But when these enzymes get degraded with time, they'll get degraded with time. So this integrity will get, there'll be some faults in the integrity of the cell membrane. Now when they will pass through the small vessels, or the small passage in the splenic red pulp of spleen, splenic red pulp, they'll get squeezed through these small vessels. And when they'll get squeezed through these small vessels, they'll get ruptured very easily, because their integrity is already compromised because of the degradation of the enzyme. And how they get degraded? They get degraded along with time. As we know, 120 days is the life of RBCs. Now when these RBCs will burst while get squeezed in through the venules, or the vessels, that's why spleen is also known as graveyard of RBC. Graveyard of RBC. Well, they'll get burst and they'll release hemoglobin. Now this hemoglobin will get phagocytosed by Kupffer cells in the liver and macrophages of the spleen. Kupffer cells and macrophages both are our phagocytic cells. Now they'll release iron, and this iron either gets stored up in the liver by transferrin, or in the it will reach to other tissues where it is required. Another thing that will get released is porphyrin. That we know, heme contained porphyrin. Porphyrin was getting bound to iron, then it was forming the heme. So this porphyrin will get converted to the bile pigment. So here's a chart showing how it gets converted. Fragile red blood cells, reticuloendothelial system, basically our spleen, which is also known as graveyard of RBCs. It will get converted to heme oxygenase will convert it to biliverdin. Now this biliverdin gets converted to unconjugated bilirubin. One thing that we have to remember that this unconjugated bilirubin gets gets conjugated with the help of liver. So before getting conjugated, like if something happens to the liver, there is some disease in the liver and the liver functioning gets nullified, or liver is not functioning or functioning very like it's not functioning well. So what will happen, this unconjugated bilirubin will start depositing in the body. So if the levels of unconjugated bilirubin is raised, it means we have something. The problem is prehepatic, before liver. Prehepatic means before liver. It could be prehepatic jaundice. Now if the problem is with prehepatic jaundice, and one thing that can happen is if there's something hepatic, what will happen then unconjugated plus conjugated, both kinds of bilirubin will rise in the level in the case of hepatic. Now conjugated bilirubin, when it will rise, when the problem is post hepatic. Then these levels will rise. Then with the help of bacterial action, this conjugated bilirubin, this conjugated bilirubin will enter in the intestine, and with the help of bacteria present in the intestine, it gets converted to urobilinogen. Urobilinogen का छोटा सा amount can get back to the liver, and to the kidneys. Then it get converted to stercobilinogen. Stercobilinogen on oxidation forms stercobilin, that is excreted out through feces. Now what happens to this urobilinogen? Urobilinogen goes to kidneys, and through kidneys it enters to urine. On oxidation, that forms urobilin, that gets excreted out in urine. So that's the end. So what happened? It formed biliverdin, then it get converted to unconjugated bilirubin, and liver conjugated the bilirubin. It formed conjugated bilirubin that gets transported to the intestine, and with intestinal, the bacteria is present in the intestinal fluid or the space, converted this conjugated bilirubin to urobilinogen. Then urobilinogen gets converted to stercobilinogen. Some amount of urobilinogen go back to liver and to the kidneys. So that's the end of this. Thank you.